Welcome to another presentation with information you'll find nowhere else. In this video, we'll primarily be talking about NAFLD, non-alcoholic fatty liver disease, and NASH, non-alcoholic steatohepatitis. But, as I always say, these things don't happen in a vacuum, because these two conditions are the liver manifestation of metabolic syndrome. In fact, the number one cause of death for those with NAFLD is cardiovascular disease, not liver failure, as you may guess. And of course, there's a microbiome component to liver health as well. If you're familiar with my videos, you probably know that it drives me a little crazy when the conventional medical establishment targets in on one organ. For our purposes here, it's more than the liver. As I always say, it's all connected. Now, the hallmark of NAFLD is a lipid accumulation in the liver. However, mitochondrial dysfunction is regarded as one of the hallmarks of NAFLD as well. And insulin resistance is considered the principal risk factor for NAFLD. But also, quote, it has been well recognized that inflammation is the key driving force of NAFLD to NASH progression. How are these all tied together? Now, there are no approved pharmacologic therapies for NAFLD and NASH, but in fact, weight loss and dietary changes have been proven to be effective approaches. But if it's so simple, then why is it that almost 25% of all people have NAFLD to some degree? Part of it is a lack of physical exercise, but you already knew that. Many people don't want to put in the hard physical work. Instead, they want magic in a bottle. Part of it is a dysbiotic microbiome driven by antibiotics, a bad diet, and PPIs, which those PPIs are meant to treat the symptoms of the previous two. And speaking of diet, some are willing to follow a diet to alleviate NAFLD, but some aren't. And to continue on the theme of diet, another part of the equation is that we are simply hardwired to be this way. Do you think it's an accident that we love fats and sugars so much? And when you combine the two of them into something like, say, ice cream, it's just amazing. This is conserved in evolution. In our history, high fat and sugar sources were rare things. They represent a dense source of fuel for an organism which may be in desperate need of energy. Thus, we are hardwired to crave these things. But we no longer live in that world. We spend too much time inactive, and we have a constant exposure to energy-rich foods, and these foods are at times manipulated to addict us. And the manipulation doesn't stop there. The powers that be in the system mislead you for the sake of their own personal interest, and at the expense of your health. But for now, think of yourself as a bear, not a lemming in the system. You want to hibernate for the winter, but in order to do that, you need to store up energy to last you through those brutal months. You need high-calorie foods to pack on the fat. Have you ever seen the documentaries on bears in Alaska? When the salmon are plentiful, the bears just eat the skin from the salmon. Why is that? Because it's a calorie-dense source of food, more so than the muscle tissue. And in the fall, there is also the ripening of fruit, which can supply simple sugars to include fructose in the diet. And as you will learn, fructose favors energy storage and moves the body away from efficient mitochondrial energy metabolism. So, that's what many people have effectively become, bears prepping for winter. But we do it for 12 months of the year. Our food system supplies us with the things that we are wired to crave, and our modern lifestyle spent on our asses doesn't do us any favors. So with that in mind, we'll be touching on the following. We'll first start with some anatomy and physiology. That will lead us into a conversation about bile acids, a topic I mention frequently. Bile acids are tightly linked to the microbiome, and they're also linked to liver inflammation. And so therefore, our third section will be on the connection between the microbiome and NAFLD. Lastly, we'll spend time on fructose in the diet, and that will further connect some dots. Again, it's all connected, and I hope by the end of this and every presentation, you'll understand that. When you think of the liver, you probably think about it as the filter of the body, removing toxic compounds, something you learned long ago, and that's very true. But the liver is incredibly active and plays roles in so many aspects of which you are not aware. For example, it makes bile acids. Okay, so those are just to help with fat absorption, right? What's the big deal? Well, they are powerful hormones which link many different parts of the body. And the microbiome plays a huge role in determining the composition of these bile acids. We could say so much more about the liver, but to dial in for our purposes here, we'll highlight its direct connection to the gut. Now, when we think of veins, we think of the circulatory system returning blood to the heart. But there is one exception to this rule, what's called the portal vein, which brings blood along with a slew of actors directly from the gut to the liver. This portal vein is a highway for our three main topics: fructose, bile acids, and components of the microbiome which dump into the liver. And when it comes to bad bacteria, the liver needs to be hypervigilant because it's pretty much the second line of defense after the gut lining, which is why approximately 15% of liver cells are Kupffer cells, which comprises the largest tissue-specific population of macrophages in the body. But this huge potential immune response may not only be geared towards bad bacteria, but can be stimulated by various triggers, which is of concern considering this immune-driven inflammation can result in disease progression to NASH. So here is our portal vein again, and one of the key components it returns to the liver are the bile acids, of which roughly 95% are returned via the ileum, while 5% continue on into the colon, where roughly 50% of the CDA is recovered, and the rest are lost in the feces. This is what's called intrahepatic circulation. It happens several times a day, and depending on your microbiome, higher or lower amounts of bile acids are lost to the feces. The liver recovers these losses by making more bile, which is from cholesterol. This is the thinking for our bile acid sequestrants, to reduce your cholesterol levels by increasing excretion. But this is only a tiny piece of the story. A small amount of bile acids escape hepatic recapture, and they reach the peripheral tissues via systemic circulation. These plasma bile acids circulate bound to plasma proteins, mainly albumin and lipoproteins, where they may exert signaling functions on peripherally expressed bile acid receptors. Keep that factoid in mind as we progress. Here we have a simplified but valuable explanation of bile acid metabolism by the microbiota. Primary conjugated bile acids, which are made in the liver or recovered from the ileal reabsorption, are released into the duodenum. But this is not the full story, because not only do the primary bile acids get reabsorbed in the ileum, but the secondary bile acid DCA gets reabsorbed in the colon. Thus, the composition of what enters the duodenum in response to a meal can vary depending on the microbiome. In the healthy, proper proportions of the conjugated primary bile acids and deconjugated, which means the amino acids are removed, enter. However, in dysbiosis, this process is inhibited, as the 26 to 29% of the microbiota who possess the BSH enzyme have been significantly reduced. Following the deconjugation, we now have CA and CDCA. The healthy microbiome further acts on these two primary bile acids via the enzyme 7-alpha dehydroxylase. However, here is an even more challenging choke point, as only a tiny fraction of the microbiota possess this enzyme, coming from the genera Clostridium and Eubacterium. To make things worse, antibiotics do a good job of killing off these few good actors. In the healthy, we have adequate amounts of the secondary bile acids DCA and LCA following this conversion. In the dysbiotic, this balance is shifted from fewer secondary bile acids to more primary bile acids, and this has many consequences. Bile acids have huge effects within the GI tract and within the liver, as you can imagine. But remember, some escape the liver and impact other organs. In addition, the organs talk to each other in a coordinated effort at survival. Here we will highlight two key receptors, the FXR and the TGR5. I'm sure these names are new to you, and we're not going to dive into the science. We already have plenty to tackle. But just know that bile acids are hormones, which means they are produced by one organ, travel to a distant location, plug into a receptor outside their origin, and kick off a series of events. Bile acids do this with many receptors, But two key ones when it comes to metabolic health, and that's what we're talking about here, are the FXR and the TGR5 receptors. These have huge implications on metabolic parameters. For example, both FXR and TGR5 are expressed in pancreatic beta cells, where they positively regulate synthesis and secretion of insulin. Well, that's interesting, isn't it? The pancreas has receptors for bile acids. That's something we associate with the gut and the liver. When bile acids plug into these two receptors, they also alter lipid metabolism for the better. They also reduce inflammation. And TGR5 even increases GLP-1. If you saw my presentation on type 2 diabetes and obesity, you may remember that the GLP-1 receptor is how Ozempic works. You may even remember that our good friend butyrate plugs into GLP-1 and kicks off the same benefits. Well, here we have bile acids initiating the same signal. But which bile acids are we talking about? Take a guess. The secondary bile acids. When it comes to TGR5, the two most potent bile acid stimulators are the two main secondary bile acids, LCA and DCA. But LCA is not absorbed well in the colon, so what we're talking about is DCA. And the DCA, TGR5, GLP-1 signals not only increase insulin production, but also sensitivity. In addition, it increases activity of thyroid hormone by increasing conversion of T4 to T3, thereby stimulating energy expenditure. You can see in this figure here, DCA with an impact on the immune system, fat cells, and muscle tissue. We see here the schematic for the secondary bile acids impacting the two key receptors and ultimately the pancreas. All of these examples are key for liver and metabolic health. So let's take a look at this bile acid information at play in a paper with 86 subjects. These researchers analyzed the plasma bile acids in those with liver disease as compared to controls, and also compared that bile acid data to liver disease status by biopsy. Before we get into the findings, let's review the bile acids because it's complicated. Here are our primary bile acids. Ca equals cholic acid, and CDCA is chenodeoxycholic acid. The G and T in front of them stand for the amino acids taurine and glycine. They get added onto the bile acids in the liver. Normally, these amino acids are removed, a process called deconjugation, by the enzyme BSH, bile salt hydrolase, by these bacteria here. But in dysbiosis, this process is disturbed. Instead of Ca, cholic acid, moving down the GI tract, we get the still conjugated TCA moving down. Normally, the next step would be to convert the primary bile acids, Ca and CDCA, into secondary bile acids. But again, in dysbiosis, especially in this step, where just a handful of bacteria are responsible for this conversion, as they possess this crucial enzyme, 7-alpha dehydroxylase, we are left with too many conjugated primary bile acids, TCA in particular, which is the main culprit in the ultimate case of dysbiosis, a C. diff infection. So what did these researchers find? Here we're looking at primary conjugated, primary, and secondary bile acids in the blood. Remember, secondary is good, primary is bad. In figure B, we see the controls had the lowest levels of primaries in the blood, while in NASH, the more severe case of liver disease here, they had the highest, far higher in fact. This was true for both primaries and both amino acid conjugates. In figure C, we see the controls with the most secondary bile acids, while NASH had the least. And in figure A, when we look at the ratio of primary deconjugated bile acids, the ratio of cholic acid, Ca, the worst of the two bad guys, over CDCA, chenodeoxycholic acid, we see once again the NASH subjects with the worst profile. This is a function of dysbiosis. Now, when we literally dive in and take a look at the health of the cells in the liver, we get this data here. Here we're looking at taurocholate, primary bile acids, and secondary bile acids. In figure A, we see as NAFLD activity worsens, total primary bile acids increase. In figure D, we see as the unconjugated primary bile acid taurocholate rises, hepatic steatosis increases. And in figure F, we see as the ratio of secondary to primary bile acids increases, a good thing, the stage of fibrosis decreases. And who is responsible for this bile acid metabolism? Your microbiome. It's actually a two-way street because your bile acids also influence your microbiome as well. So bear with me for a moment. If on a daily basis we are excreting 20 to 80% of our bile during its enterohepatic circulation, then what will comprise the bile if we have dysbiosis? If Ca and CDCA don't get transformed into secondary bile acids, then our bile acid pool will consist of a higher portion of primary bile acids. If they do get transformed into secondary bile acids, then the DCA, which is efficiently absorbed from the colon, will comprise a higher percentage of the bile. Now, consider the SIBO fans. DCA, the secondary bile acid from Ca, is the most potent antimicrobial bile acid. And to take things a step further, deconjugated bile acids, when the taurine and glycine are removed from the primary bile acids, they too are more potent antimicrobials than the conjugated form. And what is SIBO? It's IBS of the small intestine. Too many bugs of the wrong variety in a location where they should not be. So if you are dysbiotic, you will have more of the conjugated primary bile acids being secreted from your gallbladder instead of the more powerful antimicrobial deconjugated secondary bile acid, DCA, absorbed from the colon. And not only is a healthy microbiome going to impact your bile acid metabolism, it's going to do many of the great things I talk about in all of my videos. These things which help out all aspects of your health, but for our purposes here, the liver. Again, one thing we're talking about is a reduction in gut permeability. See that presentation for more information. This means less of a pathogenic load making its way to the liver within and outside of the portal vein. It also means less inflammation via butyrate's effect on T regulatory cells, something I covered in my videos on autoimmune disease. Increased butyrate also increases GLP production and GLP receptor expression. GLP is that pathway of how Ozempic works. See more on that in my video on type 2 diabetes and obesity. So a healthy microbiome supports your liver health while a dysbiotic one burdens it. And if the microbiome is dysbiotic, whole bacteria, parts of bacteria like LPS, too many conjugated primary bile acids, food proteins, and possibly too much fructose work their way directly into the liver to burden it with a huge load of factors to drive liver disease. If your diet is bad, that drives dysbiosis, which drives liver inflammation. If you have too much fructose in your diet, as you will learn, that drives liver inflammation, fat deposition in the liver, and cellular death. If you take PPIs to help with your bad diet, that too drives dysbiosis. See my video on PPIs. If you have taken too many rounds of antibiotics in your life, that also drives dysbiosis. See my presentations on both antibiotics and C. diff infections. If you're familiar with my videos, I mention LPS very often. It's a part of the cell wall component for gram-negative bacteria. Now, not all bad bacteria are gram-negative, but quite a few are. And although LPS is very immunostimulatory, LPS from E. coli is far more problematic than, say, LPS from Bacteroides. Nevertheless, it's a good marker to gauge gut permeability and inflammation. Why is that? Because our cells contain a warning system called toll-like receptors. They are an alert system that screams, alarm, alarm, when it detects invaders inside the castle wall. And a particular kind of toll-like receptor is called TLR4. This one is geared to sense LPS. And when it does, it sends a set of messages to the immune system, which results in an inflammatory immune response. Just a brief pause here in the presentation. If you could just hit like and subscribe, it would really help this channel out. As you can imagine, you want to summon the troops to fight off the invaders. And this is good in the short term. But we're talking about chronic inflammatory conditions in my videos, of which NAFLD is one. This long-term hyperinflammatory response is a bad idea. This leads to disease progression into NASH, where 20% of all NAFLD patients will go, and possibly further to cirrhosis and beyond. There comes a point when reversing the disease becomes more challenging. With that said, let's address the microbiome sooner than later, so we can avoid this process of dysbiosis, gut inflammation, gut permeability, bile acid dysmetabolism, and liver inflammation. Because we know for sure that there is dysbiosis in liver disease. How do we know that? Do you remember zonulin from my videos? That thing we make in response to gut inflammation, which causes intestinal permeability? Here in Table 3, we see significant increases in gut permeability in liver disease as compared to healthy controls as measured by plasma zonulin. But we also see far higher permeability as the severity of liver disease increases. And here's another example. In this study, 40 NAFLD patients gave both blood and liver biopsy samples. Among the blood markers were LPS binding protein, again something I reference. LPP is yet another thing the liver does. It makes it in response to the inflammation driving LPS. LPP plays a role in disposal of LPS, which we as humans are very sensitive to. Therefore, LBP is an indirect marker of gut permeability caused by dysbiosis. Well, here in Figure 1, you can see that those with NAFLD had significantly higher levels of serum LBP than the healthy controls. And as the severity of liver disease worsened, so did the levels of LBP. For more on these markers and gut permeability, see my presentation entitled Gut Permeability, the Link Between Gut Dysbiosis and Systemic Disease. Also in Table 2, we can see that the inflammatory cytokine, TNF-alpha, was significantly higher in liver disease in both plasma and liver tissue and worsened with disease. This is a marker of inflammation which can be tied to dysbiosis from the gut, reaching the liver via gut permeability or via the portal vein. And in their Figure 3, you can see the clear correlation between LBP and levels of inflammation within the liver tissue. To look at the whole picture, we can reference meta-analyses like this one. They included 15 studies in this paper and found that, quote, changes in the abundance of Escherichia, Prevotella, Streptococcus, Carpococcus, Faecalibacterium, and Ruminococcus with the universal intestinal bacterial signature of NAFLD. Well, in my meta-analysis, I included 44 studies. I've put this together like all my others for every other major condition/disease I've analyzed over the years as the Director of Medical Education for a microbiome firm and now with my own platform. Here we have the microbial fingerprint for NAFLD. These data points are from human fecal microbiome papers comparing those with NAFLD and NASH with healthy controls. As always, green signifies significantly higher in the healthy controls, while orange signifies significantly higher in the liver disease cohort. As you can see, we have many familiar actors. Promoters of health include Narostipes, Carpococcus, Eubacterium, Lactospira, F. prausnitzii, Adoribacter, Oscillospira, Roseburia, and Ruminococcus. While the classic opportunistic pathogens E. coli, Klebsiella, Streptococcus, and Bayonella are always or almost always significantly higher in liver disease when a significant difference is found. I put negative signs above the taxa, which are gram-negative. These are the ones which produce the LPS I mentioned so often. As you can see, they're associated with disease. As always, I try to highlight new and interesting information for you. Here, we have a couple of slides on the connection between the dysbiosis we just saw and endogenous, that means created from within your own body, alcohol production. Now, with bacteria, we're talking about fermentation. Some results of fermentation are highly beneficial, like butyrate. Others, like from protein sources, like P. cresol, are harmful. More on that in a later video. And in some bacteria, their fermentation end product is alcohol. Now, alcohol is a normal byproduct in the gut. This alcohol is immediately and almost completely removed by intestinal and liver alcohol dehydrogenases. But when these enzymes are inhibited and or alcohol production is elevated, blood alcohol levels increase. This alcohol generation goes on to contribute to liver inflammation, contributing to NASH, which kind of takes the non-alcoholic NA out of NASH. But what's going on here? Is there more alcohol production, or is something happening in the function of the enzymes that metabolizes alcohol? The answer is a bit of both. Here in this study, the composition of gut bacterial communities of NASH, obese, and healthy children was determined. You can see in D that the NASH children had far higher levels of E. coli, the poster child for bad actors in the gut, as compared to normal and obese children. And in figure 4, you can see that their blood alcohol levels were also significantly higher. This difference was attributed to one strain of E. coli in particular. But given the numbers I saw, it appears the NASH children had a more difficult time metabolizing the alcohol production from this fermentation. If we look at a separate study in adults, we see strains from a different species, Klebsiella pneumoniae, as the cause of the endogenous alcohol problem. To validate the correlation between high alcohol-producing Klebsiella pneumoniae and NAFLD, they analyzed the abundance of K. pneumoniae and alcohol production in individuals with NAFLD, including non-alcoholic fatty liver, NASH, and healthy controls. Although the abundance of K. pneumoniae was slightly higher in the feces of NAFLD patients than in healthy individuals, the alcohol-producing ability of K. pneumoniae was significantly stronger in the feces of NAFLD patients, which you can see in figure 1E and 1F. The data also showed in figure G that 61% of NAFLD patients carried the high alcohol and medium alcohol producing K. pneumoniae strains, while this value was only 6% in controls. When they looked at mitochondrial dysfunction, they found a lot. Remember, the mitochondria are the powerhouses of the cells by producing something called ATP, the basic unit of energy. If your mitochondria aren't healthy, you've got big problems. When it comes to liver health, mitochondrial dysfunction plays a pivotal role in the development of NASH, as it not only impairs fat homeostasis in the liver, but also leads to the accumulation of reactive oxygen species, think oxidation, which then triggers lipid peroxidation, cytokine overproduction, and cell death. Considering the alcohol-generated damage from these Klebsiella species, these researchers analyzed the mitochondria for abnormalities to include mitochondrial integrity, ROS accumulation, that's reactive oxygen species, membrane potential, lipid peroxidation, ATP levels, and mitochondrial DNA damage, all showing clear mitochondrial injury. And not shown here, they also measured the blood alcohol content in NAFLD patients, NASH patients, and healthy controls after an oral glucose tolerance test. The data revealed that the mean blood alcohol content of NAFLD patients was significantly higher than that of the control group, especially in individuals with NASH with a higher weight. The link between healthy mitochondria, alcohol metabolism, and the slides we'll be covering next can be summed up with 3 letters, NAD. I always talk about how butyrate is one of the most important compounds for health in our bodies. Well, here's the other, NAD, and it's absolutely crucial. With DNA repair, energy production, sirtuin metabolism, which is the whole resveratrol, longevity, and prevention of disease thing, and so much more. It also plays roles in alcohol metabolism. So if your NAD is depleted, your ability to metabolize alcohol is reduced. And likewise, if you drink alcohol, you deplete your NAD stores. Alcohol is known to rapidly generate uric acid. If you have gout, you know what uric acid is, and you know you should avoid alcohol. But uric acid isn't just for gout and kidney stones. It's pro-inflammatory. High uric acid in your blood is closely related to the onset and progression of many diseases, such as diabetes, metabolic syndrome, chronic kidney disease, cerebral vascular-related diseases, high blood pressure, and hyperlipidemia. Studies have confirmed that uric acid level is an independent risk factor leading to the progression of cardiovascular-related diseases, such as hypertension and coronary heart disease, and has an important impact on its prognosis. And remember that the number one cause of death in those with liver disease is cardiovascular disease. With that in mind, we look at a microbiome study in those with hyperuricemia, HUA, high uric acid in the blood. Now, healthy people excrete uric acid in two ways, of which 70% is excreted through the kidney, and the remaining 30% is excreted through the intestine. And your microbiome affects the metabolism of uric acid, both directly but also indirectly, by reducing the burdens or oxidation and inflammation on the liver, not to mention the kidneys, but that's a later video. Once again, we see some class results. Health promoters significantly higher in the healthy controls were species from Coprococcus, Eubacterium, Butyrosamonas, Ruminococcus, Allostypes, Adoribacter, and Roseburia, to also include species C. leptum and Eosinophylla, while bad actors significantly higher in those with high uric acid were species from Fusobacterium, R. torques, and a bad actor I frequently mention, R. navis. Keep this color code here in mind, which signifies purple is positive and blue is negative. So if we zoom in and focus on the leftmost column called SUA, which means serum uric acid, we see purple up top, positive association, and a lot of blue, negative association on the bottom, which means that the more Fusobacterium species and R. navis in the microbiome, the more uric acid in the blood. On the flip side, we see when there were more of the health promoters I previously mentioned, there was significantly less uric acid in the blood. So now let's take this uric acid story one step further. The consumption of high fructose corn syrup, which is ubiquitous in so-called foods, causes a spike in uric acid formation, and many studies support this. So let's look at the biochemistry. The metabolism of fructose is different from that of glucose or the other sugars. In fact, fructose is the only carbohydrate generating uric acid during its metabolism. Fructose is absorbed and literally dumped into the liver via the portal vein in a somewhat unregulated fashion. This enzyme, which first metabolizes fructose, is called the fructokinase, and it makes this metabolite here. However, this is very energy intensive. If you recall, ATP is the basic unit of energy, which we see here. This is rapidly consumed by the enzyme fructokinase. You can see here that there is one phosphate group on fructose, but here there are two on fructose 1,6-bisphosphate. That phosphate came from the ATP here. ATP stands for adenosine triphosphate. When one phosphate is removed, it becomes ADP, adenosine diphosphate. When two are removed, as seen here, it becomes AMP, or adenosine monophosphate. The presence of AMP is a stress signal of energy depletion, and another enzyme degrades AMP ultimately to uric acid. This is bad for several reasons. The depletion of ATP in the liver cells has enormous consequences. In addition, the uric acid itself is a big problem. It drives free radical production, inflammation, and even cell death. We don't need any more inflammation in cells battling too much lipid content, bile acid imbalance, and toxic components arriving from the microbiome. As for the connection to blood sugar, studies show as insulin goes up, urinary excretion of uric acid goes down, a cycle of deterioration. Furthermore, high fructose intake with consequent adenosine triphosphate depletion and uric acid formation decreases fatty acid beta oxidation, which is the burning of fat for fuel, which leads to hepatic lipid accumulation. And speaking of lipids, we know that there is an accumulation of lipids in the liver and NAFLD, and this leads to lipotoxicity and liver damage, potentially progressing as far as liver cancer, as shown here. We also know that fructose raises triglycerides. We also know that fructose induces visceral fat accumulation, dyslipidemia, insulin resistance, impaired postprandial fat oxidation, and reduced resting energy expenditure. Furthermore, increased fructose intake in humans has been linked with progression of liver disease to fibrosis, while fructose restriction leads to improvement in NAFLD. Additional studies have found that fructose consumption is increased nearly 2 to 3-fold in adults with biopsy-proven NAFLD. So what's happening in the cell biochemically to generate this high lipid environment? Well, as mentioned previously, the powerhouse of the cell is the mitochondria, as shown here. Now think of it as an engine, like your car or your lawnmower. If you flood your lawnmower with gas, what do you have to do? You have to keep pulling on the cord until you've burned off the excess fuel inside so it can run properly. The mitochondria aren't much different. When functioning normally with that all-important molecule NAD I mentioned earlier in alcohol metabolism, that unit of energy, ATP, is produced efficiently, and we have the energy to do the things we want. But when you flood it with carbon inputs, in this case pyruvate from high fructose corn syrup, it floods the engine, and it kind of freezes until it can purge the extra carbon inputs in the form of citrate by exporting it into the cytoplasm of the cell. But the citrate doesn't magically disappear into the air like the excess gas from the lawnmower. It undergoes a series of steps, which results in triglyceride synthesis and accumulation in the liver cells, which we established at the beginning is the hallmark of NAFLD. This process is called hepatic de novo lipogenesis, which means the making of lipids in the liver from scratch. And it is well established that de novo lipogenesis is far higher in NAFLD than in normal livers. And as the blue box states, quote, fructose metabolism supports de novo lipogenesis more strongly than a high-fat diet. That's kind of a shocking statement. You'd think dietary fats would drive fatty liver more than anything else. But that's just one of the many things you'll learn here. Now, I'm not saying that dietary fats are great for NAFLD, especially saturated fats. There is plenty of data to show that saturated fats are a bad idea. But we can't tackle every aspect in one video, so we'll stick with fructose. And not only is the fructose directly overwhelming the machinery of the hepatocytes, the liver cells, but it also sends signals to powerful hibernation pathways. I always talk about signals. Well, it's no different here. These two guys here get upregulated. They play powerful roles in the liver and beyond in this hibernation process. And guess what represses their expression? FXR, that important receptor from earlier, which is upregulated by the secondary bile acids. And also insulin is a powerful player in this equation, which also stimulates this thing called SHRB1. Now, although fructose, unlike glucose, does not require insulin for its metabolism, chronic fructose ingestion can lead to hyperinsulinemia and insulin resistance. And there's more. Do you have high blood pressure to go along with your obesity, liver disease, and high blood sugar? Well, many studies show fructose increases blood pressure. This mechanism probably involves one or more of the following: stimulation of salt reabsorption in the kidney, increasing serum vasopressin levels, and the production of uric acid, which in itself we saw is pro-oxidative, which affects nitric oxide production, which is important for men with erectile dysfunction. More on that in my video on cardiovascular disease. Fructose metabolism also drives the development of central leptin resistance, resulting in persistent hunger and or impaired satiety, which results in exaggerated food intake. This goes back to the beginning of how I talked about how we are wired for certain rewards. This fructose metabolism is how we get papers like this entitled, quote, serum uric acid concentrations in fructose consumption are independently associated with NASH in children and adolescents. By the way, pediatric NAFLD is associated with a 14-fold higher mortality risk. But why are U.S. adolescents allowed to ingest, on average, 94 grams of added sugar per day, 20% of their energy intake? Much of this coming from high fructose corn syrup added to food and drinks. And when we say high in the fructose corn syrup, we're not kidding. When we think of bears eating apples for its easy supply of energy for winter hibernation, you have to realize that a ripe apple is only 5 to 10% fructose. High fructose corn syrup starts at 55% fructose and can go as high as 90%, which makes it sweeter than table sugar, which is 50% fructose. The high fructose corn syrup story got started in the mid-1900s when, following the World Wars, sucrose became scarce. After decades of effort, the sugar industry, in all of its benevolence, brings us disease with a smile, which we happily purchase. And speaking of the sugar industry, you would think with what you have just learned, this would be common knowledge, that someone from a government agency would be looking out for you. Far from it. Don't trust the system. They do not have your interests in mind. They only have theirs in mind. You see, farmers have known for centuries that feeding corn to animals resulted in weight gain. And for decades, while we've been waging a war on fats, the world has only gotten fatter. Think about it. Decades of low-fat this and no-fat that has resulted in enormous gains in obesity and all the diseases that go with it. I'm not saying saturated fat is good for liver disease. There is a ton of evidence to prove that it's bad. But arguably, sugar is worse, and especially high fructose corn syrup. But this too has been known for decades, just hidden. I recommend you read this paper. It talks about the lies and corruption on many levels. The idea being to villainize fats and downplay the evidence behind sugar's role in lipid metabolism. All done at the hands of the Sugar Research Foundation. You can read just a few quotes from the paper here. This was all back in the 1960s. The absolute corruption in the U.S. at every level of decision-making and influence has only gotten more disgusting over the decades. So it's time you educate yourself. You need to take charge of your health care. I provide you with many references. You can easily find and read all of these papers if you wanted to. There are so many lies and places where you can go wrong. I try to uncover as many as I can throughout my videos. When it comes to liver health, you should now have a better understanding of what to do. Yes, there are dietary considerations. Yes, there are dietary supplements which can address the root cause. With the proper guidance, they can help address the dysbiosis, inflammation, gut permeability, and leakage of undesirable gut contents in amount and type into circulation, and via the portal vein directly to the liver, driving inflammation. There are supplements which can address the reductions in NAD, which can help with uric acid, inflammation, mitochondrial health, sirtuin metabolism, oxidative stress, energy levels, and so much more. And there are supplements which can help with bile acid metabolism as well. And you don't have to reinvent the wheel. Luckily, you have a microbiome expert who is ready and able to help, and whose goal is to get you better as quickly as possible, as opposed to a system which profits off of your illness and suffering. Well, I hope you enjoyed that video, and I will say that basically every single day on my YouTube comments and in emails, I get a lot of thanks from people out there who really appreciate the information I've been sharing. And so part of that is you're welcome. The other part is you can contribute by clicking on the super thanks below. And if you're not doing a consultation with me and you're not purchasing any protocols, it's a great way to support this channel. Each presentation, depending on the presentation, but most of them take an incredible amount of time to put together. There's a lot of material. There's a lot of data checking. And so it's just sometimes 50, 60, 70 hours to put together one presentation. And so if you can just click that super thanks, I'd appreciate that, and we'll keep the information coming.